Good morning, everyone. I first want to thank Dr. Richard Knight for giving me a kind introduction, and once again, thank the Foundation for giving me opportunity to give a report of our effort trying to develop a comprehensive approach for effective prevention and treatment of human prion diseases. Prion disease, as many of you know, is a group of fatal transmissible disease that requires the cellular prion protein, or PrPC. Unfortunately, there's no treatment available at this point. Purification not only requires PrPC, but also requires PrPC as the mediator or receptor for its toxicity. And more importantly, or equally importantly, PrPC appear to be dispensable because in knockout mice or other animals knockout with PrPC knockout, there appear to be no big problems. So this makes PrPC the ideal target for therapeutic intervention. Cellular PrP is a small protein. As shown here, an N-terminal part is a flexible region of flexible tail. C-terminal part is structured globular domain, and normally goes through different kind of processing or cleavages. I want to highlight the outer cleavage, which cut almost the middle of the protein, which will destroy the capacity of the PrPC as a substrate for purification. In the meantime, produce two beneficial fragments, N-terminal N1 and C-terminal C1. N1 has been shown to be neuroprotective. I'll talk about more later about it. And the C1 has been shown to be able to inhibit purification. Prion disease is complicated and diverse. In humans, the primary form is CGD, which can be subclassified into sporadic form and familial and acquired forms. The sporadic form also has multiple subtypes. So this because there are other types of human prion disease. This huge complexity and diversity among human prion disease patients is a huge challenge when you try to develop a therapy against all human prion disease. So the efforts to develop a therapy against prions can target different aspects of the prion replication, clearance, or pathogenesis. Basically, it can try to reduce the cellular PrP level, which is the substrate, the PrP substrate, and also receptor for toxicity. And second, it can try to inhibit the prion agent PrPSC replication. It can also try to enhance the clearance of the PrPSC, or it can prevent toxicity presented by the toxic molecules during prion pathogenesis. And it also can try to restore or regenerate the loss of neuron or neuronal functions. And finally, it can try to inhibit neuroinflammation that has been shown to be important in prion disease pathogenesis. So how to achieve those goals? You can do either by small molecules, which is commonly used by most pharmaceutical companies, try to affect any of these processes. Secondly, because anti-PrP antibodies, which will try to knock down PrP level or clear PrPSC. And you can also use the siRNA, which will try to knock down mRNA encoding the PrP gene to reduce the PrP levels. The same goal can be achieved by using antisense oligonucleotides, which has been shown great potential in animal models studied recently. And you can also use stem cell therapy to replenish the lost neurons or other brain cells, try to restore function. And you can also use gene therapy to achieve any of those goals here. And finally, you can try to develop a vaccine that will prevent or eliminate the prions before or during its replication. However, there are quite a few drawbacks of the current efforts up to this point. First, most studies have been done using non-human prions, basically use animal prions, and human prions may be different. And second, they usually only target one or two aspects of the prion pathogenesis. Unfortunately, as I told you before, none is proven effective in clinical trials so far. And even if it works, most of the strategy will also require frequent and long-term dosing. So I'm trying to develop a comprehensive approach that will reduce the PrPC level, attenuate the PrPSC level, and protect against neurotoxicity at the same time. And then we're going to use human prions, basically CGD prions, and studied them in human neuronal cell line or humanized mice expressing human prion protein, so that the result can be more easily translatable to clinic if it works. So there are three elements to our approach. We call it RAP. First is use doxycycline, which are given orally. Doxycycline is an antibiotic that has been shown to be effective in prion animal model studies. And in clinical trials, it's safe, but did not show protective effect, unfortunately. And the second element is using anti-PrP messenger RNA constructs to knock down PrP level in the brain or in the central nervous system in general. Let me explain what RAAV is. RAAV is a recombinant adeno-associated virus, which is the commonly used viral vector for gene therapy experiments and for some clinical approved gene therapy treatments. It is good because it's safe and it can achieve long-term expression after one shot, and it can be also made into plasmids that can be used as a plasmid therapy if you have some other reagent to help plasmid DNA get into the cell effectively. That's why we choose recombinant AAV vector for our experiments. And the third is using the expression of a secreted form of the neuroprotective PrP N-terminal fragment, which will also be mediated or delivered just like the enhanced PrP and shRNA using a recombinant AAV adeno-associated virus vector, which has been shown to be the safest viral vector so far for gene therapy. And the last element, which is not directly associated with the study, but we are trying to develop novel nanoparticles, which will enable safe and efficient in vivo DNA delivery to achieve gene therapy, to bypass the need for viral vectors, which have been shown to be very expensive and carry other safety risks. So in terms of reducing the PrPC level, we'll use several anti-human PrP siRNA constructs that we carried on the recombinant AAV vector. And recombinant AAV siRNA vector will be either made into viral particles, which is the traditional way to do it, or we can directly use the naked plasmid DNA and carry this siRNA and use them in conjunction with our novel nanoparticle reagents to deliver them efficiently into the brain or spinal cord. Then these RAV vectors will be introduced in the brain or spinal cord, the space where the cerebral spinal fluid can flow through the entire central nervous system through the intracerebral ventricular or intrathecal infusion. This way is to bypass the blood-brain barrier so that they can reach the nervous system tissue to achieve its effect. To attenuate reduce the PrP assay levels, we achieve it through three ways. One, as I said before, is use oral administration of doxycycline, which has been shown to directly inhibit prion replication. Second, by knocking down the PrPC level using shRNA, we'll also knock down the substrate, which is the PrPC, for prion replication. And the third, by expressing the secreted form, the N-terminal fragment of PrP, we'll show later that actually this N-terminal fragment can also inhibit prion replication. So these three ways will all come together to achieve the goal of reducing PrPC levels, PrP assay levels. So where we are on this, we have started oral feeding of doxycycline-laced rodent food to either specifically MM1 subtype or MM subtype inoculated human mice. And second, we have generated the anti-human PrP plasmid constructs, and they are being examined for the efficiency and specificity for knocking down human PrPC expression in human cell lines and in human mice. And we have shown, we'll show a little bit later, that the N-terminal fragment of PrPC can directly inhibit at least the Prion-seeded PrPC amplification in archaic acid. So in terms of protection against neurotoxicity, it will be done in two ways. One, we achieve that through recombinant AAV-mediated high-level central nervous system expression of the secreted from the PrPC N-terminal fragment. And second, it can also achieve through siRNA-mediated knockdown of the PrPC gene expression, because this will reduce the PrPC level. The PrPC not only is the substrate, but also is a mediator for prion toxicity. So why do we choose the PrPC N-terminal fragment and use that to try to use that to achieve neuroprotective effect? N1, as I showed before, is an N-terminal fragment normally produced through over cleavage of cellular PrP. It's shown a good, strong effect to prevent cell death in culture cells, and it's been shown to also bind to toxic A-beta oligomers. That is the central player in Alzheimer's disease pathogenesis, and by doing through binding, they can neutralize toxicity in Alzheimer's disease. And it may also do the same thing with the other toxic molecules, like in Parkinson's disease, maybe in tauopathy even. And the work from our collaborator, Dr. Wenchen Zhou's lab, have shown before, recombinant PrP seem to be able to inhibit prion in PMC reactions. And this effect seems to be largely reside on the N-terminal fragment of PrP. So we hypothesize that the PrP N-terminal fragment will also protect against the prion toxicity. And I'll show later that we've shown that the N1 can also inhibit prion directly in RD quick regression. So here's our RD quick data showing that if you add recombinant N1 to the prion seeds before you start the RD quick regression, it can achieve it can prolong the lag phase of the seed amplification when you have less seeds, the effect is more dominant, it is more prominent. When you have more seeds, when your seeds come in higher, the effect is still there, but it's not as dramatic when you have less prions. So before I had this grant, we actually tried a quick dirt experiment before, where we just introduced the recombinant AAV carry the N1 gene intramuscularly to wild-type mice, and then inoculate the mice with mouse Prpie, and see what happens. Unfortunately, see that the recombinant AAV N1 injected mice, they showed a slightly longer incubating time, but it's not significant. But one good thing is, at least do not make things worse. It seems to have some slight effect, but it's very uncertain there. But then we look at whether we can detect the N1 expression from that RAV N1 factor. And unfortunately, we cannot see it when you introduce that gene into the C2C12 muscle cell line. We cannot detect N1 in the medium. And this is not surprising, since after that we learned that from Alex Mippen and his colleagues, result that the direct prion N1 probably is not going to work because it's going to stuck inside the cell, cannot get outside. So to address that problem, we designed a larger N1, we call N1 plus, which we show that it can be detected if we introduce the muscle cell line, can detect it in a large amount in the culture medium. We repeated that later again in another experiment, in many subsequent experiments. More importantly, we recently introduced this recombinant AAV N1 plus construct into mice by intraventricular infusion. And we then collected plasma. Here shows that at 4 weeks after the infusion, we can detect the N1 plus fragment in the plasma of the injected mice. and it's not this same thing is not showing up in the mice that injected with control RV GLP plasmids, showing that it's a specific only when you inject the A1 plus construct. So encouraged by this result, we started 2 animal experiments. One, we're using the TGN6H mice, which express human PrP with no glycans. And we show this because it has a very short mean time with the MM2 substrand of human sport CJD. That's just a little over 2-month period. We inject these mice first with this RAV particles, RAV plasmid, with either A1 plus or the GLP control. And 3 weeks later, we inoculate them with this pre-adapted MM2 Prion strain. And now I'm waiting for the symptom to occur, then to check for their pathology and the PrPC accumulation. The second experiment we did is using the T40H mice, which expressing wild type human PrP. And we showed if we inoculate this mice with the common MM1 substrand of CJD, it has about 6 months of incubation period. Now we're injecting animal in 3 groups, 2 of them with the A1 plus construct, the other with the GLP control construct. And after that, 3 weeks later, we inoculate with MM1 Prion. And then one group will be given doxycycline 1 week later. And now we are also waiting to see whether any of the treatment have protective effect on the prion disease progression. And this is where we are at this point. We are planning to further test our shRNA construct to show their specificity and effectiveness in neuronal cell lines and human mice. And we're going to, once that's done, we're going to use the best shRNA construct to treat them, to use them to treat our mice, human mice, either alone or in combination with the A1 plus construct and the oral doxycycline. And we're also going to try to further optimize and validate our nanoparticles so that we'll be able to achieve affordable and safe, effective in vivo gene delivery to patients affected by either prion or potentially other disease like Alzheimer's disease. Finally, I want to thank the people in my lab, especially Qingzhong, Manuel Camacho, and several very talented students, and also Qi Xu for conducting this work. And my collaborator, Dr. Wenchen Zhou, and Dr. Wilder Schultz, who provided the recommended A1, and for the Prion Pathology Surveillance Center, which helped with our quick experiments. And I appreciate all the families affected by CJD, and also I want to thank definitely the funding provided by the CJD Foundation and the Strides for CJD, and the families who generously provide the funds, the Gary Butterman Memorial Grant, the Michael Cole Memorial Grant, the Ernie Donahoe Memorial Grant, and the Priest Family Memorial Grant, Research Grant, the Tom Stevenson Memorial Grant, and the Teresa A. Strauss Coolidge Memorial Grant. This funding has made it possible for me to jumpstart my long-term dream trying to develop therapies to treat human prion disease, because I see the suffering of families, and I actually been contacted by a few families who I never know of, tried to ask me for help, but I felt bad I could not help them. But I hope with this funding, with our started project, I hope in the near future we'll be able to help them. And thank you for your time and attention.